Now today we will study about principle of inheritance and variance. This topic is very important because we are dealing with genetics, and here we will understand that who is known as father of genetics. So father of genetics is known as Gregor John Mendel. He is known as father of genetics, and he has worked on Pisum sativum, which is called as pea plant, for 7 years, that is from 1856 to 1863. So he has given 3 laws of inheritance that we will study in this class. And the first law is known as law of dominance. Second is known as law of segregation, and this third law is known as law of independent assortment or dihybrid cross. So first law, which is called as law of dominance, this we will understand. Before that, we will understand some genetic related terms which is very essential to understand, because without the terms we can't able to understand the law properly. So the terms which is very essential is as follows. First term, we will see that what is gene or what is allele. Then what is trait? What is character? So we will understand that gene, allele, trait, characters, or we can talk about that factors. These all terms are same. Whether you can say gene, allele, trait, character, or factors, these all terms are same, which indicate these alleles, basically the terms which is used by Gregor John Mendel, that is allele. In some books they are using factor. Factors or allele, they comes in pair. They comes in pair. So character, one particular character is always carried with in the terms of pair. As we can say that how we'll write any, suppose we are taking one character like tall plant, that will be represented by capital T, capital T. If you are talking about a round pod, small capital R, these are dominant character, which is represented by capital letter R called as dominant character. And if the character we have to write with the recessive small letter like this, small t, or the seed like small r, so they are supposed to be written in small letters. They represent the particular character in pair. So this we have understand. Now we will move to the second important thing, which is called as phenotype. This term is very important. Phenotype, Phenotype. Phenotype is that character. Pheno means morphological character of any cross that we can see. Morphology means from outside what the character is visible is known as phenotype. The morphology, that is a phenotypic character, or we can say phenotypic ratio. The ratio we can obtain after a cross, that is known as phenotypic ratio. The next term which we have to understand, that is genotype. Genotype or genotypic. Genotypic character. Geno means we are relating with gene, and type means we are getting a ratio like gene related ratio. What is there? That analysis is done with the help of genotypic ratio. So in cross we will see that phenotypic ratio as well as genotypic ratio. Both the ratios are important to understand whether it is genotypic ratio or phenotypic ratio, right? Then we will understand dominant character as well as recessive. What is dominant? Dominant means the self-representing character is known as a dominant character. So dominant character is that character which is ruling character. Also, it is also called as a ruling character. And second, that we can suppressed character is called recessive character. Suppressed character is known as recessive character. So by this way we have understand some terms. Then one more important thing we will understand, that after cross what we are getting, offspring of progeny. They can also call as offspring offsprings you can say, or you can say progeny, or you can say they are daughter cell. So these are same thing, whether you can say offspring or you can say progeny or you can say daughter cell, all three are same that we talk about. So this is very essential thing. These are some genetic terms which we have talked about. Now first of all we will talk about, with the help of Punnett square will understand law of dominance. We are going to understand this first law which is called as law of dominance. We will see here what is happening in the case of law of dominance. This will understand, because here we are talking about what is law of dominance. This will understand with the help of a graph. In the case of law of dominance, we will see that one factor in pair of traits means character or allele, dominates the other inheritance character unless both factor in a pair are recessive. And this is called as law of dominance. This is the first law given by Gregor John Mendel in 1856. So he told that with the help of Punnett square we are taking some characters, like we will talk about two contrasting characters, like suppose dominant character and second we will talk about recessive character or traits. We will talk about, like we will talk first height of the pea plant. What Mendel has done, he has done the study about height of the Pisum sativum, that is called pea plant. So dominant character will be capital T, which will be represented with the help of capital T, and recessive character will be represented by small t. Then second we will talk about seed, which is seed. In the case of dihybrid, we will understand seed. What type of seed? Round seed, which will be denoted by the help of capital R, and wrinkled seed. Opposite of round is wrinkled, and that will be represented by small r. Then next we will talk about pod, color of pod, that will be of two types. First, when we talk about red color, that is red color, and yellow color. Two colors are there, like yellow color or green color. So yellow color will be represented with the help of capital Y, and color of the pod will be green, that will be represented by small g and small g. So these terms we are going to use in making a cross. So first of all, when the law of dominance, how these terms will be helpful. So we will take one contrasting character, that is height, and with the help of height we will show the law of dominance, that is dominant as well as recessive character. So we will understand here. In this, we used to take a parent cell. They have two colors. Suppose one parent is tall, which will be represented by capital T, and cross in between with a small t and small t. So capital T is known as tall parent and small t is known as dark means short height. So how will we make a cross? In first generation we will see, what we are going to do, we are isolating the alleles in such a manner that cross can take place easily. Small t with a small t, as you can see here, First the cross will be taken from first gamete, second cross, the second gamete, first gamete and then second gamete. So what we obtain? We obtain that capital T small t, capital T small t, capital T small t, capital T and small t. In this we understand that this is the F1 generation cross, and here we will understand that capital T when crossed with dark parent is small t, in first will all the four gametes you will get that are tall. All the four gametes that is tall. So in phenotypic we will say the ratio phenotypic ratio is 4:0. So this is the phenotypic ratio because the gametes are similar. So from morphological it is similar, all are impure tall, they all are similar, and that is why we are getting 4 gametes, and there is no difference, that is why 0 ratio. In the case of genotypic ratio, again we will see that here also we are getting the same ratio. Means genes you will see capital T everywhere, 4 capital T's are there, 4 small t as they are making a gamete in pair. So this is the genotypic ratio and this is the phenotypic ratio, that is 4:0, 4:0, and this is law of dominance because the tall character which is a dominant character and small t character which is a recessive character is suppressed by dominant character, and that is why all in first generation all the gametes are tall. Whether it is phenotypic or genotypic, we are obtaining a ratio 4:0. Now by this way, the Mendel has explained his first law of dominance. Now we are going to study the law of segregation. After this we will study the law of segregation. This is the second law. The law of segregation we will see that production of two gametes in this law of segregation, Mendel has seen that production of two gametes, the two copy of each hereditary factor, segregates. So that offspring acquired first one factor from each parent. Mean to say that in the production of two gametes, the two copies of each heredity factor segregates so that offspring acquired one factor from each parent. So how we can understand this law? We can understand with the help of flowchart. As we have already talked about law of dominance, same way we will take a gamete representation here. We have taken parent cell, that is say contrasting character about regard height of the plant in Pisces sativum, and that is represented by capital T which will be crossed by small t, and that is tall plant, and this is dark plant. Here we will write these are the tall plants, and here we are showing dark plants. As we can understand, we will isolate the gamete, we will have the cross. Here we will get a F1 generation cross. In F1 generation cross, we will see the two gametes formation as it is taken place. First of all, we will see that capital T will be crossed by small t, then again capital T will be crossed by small t, and we are going to obtain capital T small t. These are the gametes. This is the F1 generation which have seen in the law of dominance also, capital T, small t. So in this F1 generation we obtain that all are impure breed, all are tall. Now we will see the second part. Capital T, small t get crossed with capital T and small t. In this daughter cell cross you will identify the gametes production with the help of a flow chart. Here we will see that capital T, small capital T and capital T, first gamete formation takes place. Second is capital T, small t, capital T and small t, then small t, small t. These are the four gamete formation take place in the case of F2 generation. What is the ratio we will obtain? So we will obtain phenotypic ratio, and that is 3:1, means the three are tall plants and here one is dark. These all are tall plants and this is dark plant. And second ratio is genotypic ratio, and here we will obtain that the ratio will be 1 pure tall, which is indicated one plant, 2 are same type tall, impure breed 2, and 1 is pure dark. So we are obtaining a ratio 1:2:1. So this is a genotypic representation, as well as this is the law of segregation, where in daughter cell these particular gametes are obtained. Now we will talk about third law, which is called as law of independent assortment. Okay, now here we will understand third law of Mendel, that is law of independent assortment, that is also known as dihybrid cross. And in this law you will see that law of chances govern will be particular characteristic feature of the parental pair will occur in each indivisible offspring. So here we are taking four contrasting characters and we will cross in between and we try to understand what is happening here. So we have taken different four characters, like seed have a round shape of the seed, that is round, and it is denoted with the help of capital R, then small r, which is showing the wrinkled seed, WRIN, wrinkled seed it is showing, then capital Y, which is showing yellow color of the pod, and small t, small r and small y and small y character, which is showing green color. So by this way, in these four different characteristics, we will understand the law of independent assortment as well as dihybrid cross. So the law of independent assortment is only known as dihybrid cross. These are the four different contrasting characters we have taken, that is round, wrinkled, yellow, which is represented by capital Y, green, small y, these are the pod colors, and round is the shape, capital R it is denoted, wrinkled with a small r and a small r it is indicated. Now we will see how the cross take place. So for this, first of all we will write here, we will take two contrasting characters like capital R, which will be in pair, capital R, and second character is they have yellow color, capital Y will be crossed with a small r as well as a small y. So this we are getting. So one we will take from this side, that is your round, and contrasting character of this is wrinkled. So small r will take one side. This is one pair gamete which will be crossed by yellow color, capital Y, and green pod, that will write by this way. So in this, when we will make a cross in F1 generation, this is the parent cell and the generation is F1 generation. Mixing of gamete, first we will cross this, and this here will obtain capital R and capital Y. This is the first gamete. Second gamete we will see here, which we will cross here, that is capital Y small r. Third gamete here, that is capital R small y. Fourth gamete we are obtaining, that is small r small y. So here we are getting these four gametes, which is very essential to understand. And again, with the basis of these four different characters, we will make a crossword of Punnett square, and we will understand the dihybrid cross. So we will see here. We are going to make a 60 blocks where the cross we can see. First gamete that we have obtained, that is capital R, capital Y. Second gamete we have obtained in a F1 generation, that is capital Y small r. Third we have obtained capital R small y, and fourth is small r small y. In same fashion we will arrange the gamete. This is in first position. So we'll write here in first position capital R capital Y. In second position we'll write here capital Y small r. Third position we'll write capital R and small y. And last fourth is small r and small y. So these are the four gametes. You have understand which we have crossed, four characteristic contrasting parent we have crossed and obtained this gamete. We will make a square root, that is Punnett square, in such a manner as you can see here. So you can see here, these are the four lines which we are drawing between. These are the horizontal lines. Now after this we will see the vertical four lines. Yeah. This is the Punnett square of 60, and how we will make a cross we will see here. See, first of all we will see, we will write the upper capital R in all the four gametes. We will put in every each section. We will keep in every each section. So it will be dropped down, like we will write capital R, capital Y. Here also capital R, capital Y, capital R, capital Y, capital R and capital Y. Here capital Y small r, capital Y small r, capital Y small r. Here we are getting capital R small y, capital R small y, capital R small y, capital R small y. Here we are getting small r small y, small r small y, small r small y, small r small y. So it is clearly indicating that here in first what we obtained, we have written capital R, capital Y. This one in four blocks, as you can see here. Once again I am telling you, capital R, capital Y in four blocks, which is coming at down in each and every block. Then small y, capital Y small r. It is also we are writing in every block the same thing. Here we are writing capital R, small y, every block. This is the easiest method to draw this. Here we are writing small r, small y. Yes. Now this side whatever is written for gametes will place accordingly in line. So capital R and capital Y will write here in every block, every 4 blocks. Now here we will write capital Y and small r. Here we will write capital R, small y. And here we will get small ry, small ry, ry. So what we obtain? We obtain that there are 4 types of gametes. Different type of ratios we obtain. So this is the round and yellow. Round and yellow first type. This we will make a circle. This is round yellow. Round yellow. If in 4 allele, if any one allele is capital, that will be dominant. If R is capital, means it is round in 4 of them. If Y is capital, that means it is yellow color. Round yellow seed we are obtaining, right? By this way. So here we get 9 gametes. Here also Ry is capital. So similar type of gamete we are going to make a oval, enclosed in a oval. Ry here also. So how many gametes? 1, 2, 3, 4, 5, 6, 7, 8, 9. 4, 5, 6, 7, 8, 9. So we are getting what we are getting? We are getting that capital R and capital Y gamete how much? Round yellow. They are called round yellow. If any gamete having either R or this both R and Y, so that will be round yellow. So these are 9 in number we obtain. Now second we will see. Second we will see that is capital Y means yellow and small r means wrinkled, yellow wrinkled. So here also we will see yellow wrinkled. Yellow wrinkled we obtain, and here yellow wrinkled. 3 of them. So capital R, capital Y and small r. What we have to see that? We have to see that one capital Y gamete is there and 2 small r are there. If you are obtaining this one, this is called as yellow. Yellow is small r is known as wrinkled. Yellow wrinkled, W-R-I-N-K-L-E-D, and they are 3 gametes. They are 3 in number. Now next we will see. This time this is the first one. This is capital R means the round and small y means green. Round and green. Round and green is here also, and round and green is here also. It is in cloud shape enclosement. So we will write here. Round means one R capital is necessary, and then a small y whether it is in a pair. So that is called a round green. How much gametes? We are getting 3 gametes. Now one last gamete we will see, and this is small r and small y. This is small r and small y. Only one which is left. So we will write small r and small y. How much we are getting? They are known as your green wrinkled. They are called green, W-R-I-N-K-L-E-D, because both are recessive, and that is why they are 1 in number. So what is the phenotypic ratio? Phenotypic ratio. Phenotypic ratio we obtain, that is 9 ratio, 3 ratio, 3 ratio, 1. Mean to say this is the phenotypic ratio. Genotypic is a big ratio which is not asked in examination, but we can see that phenotypic ratio we are obtaining 9 means round yellow. How many seeds are there? There are 9 seeds which are round in shape and they are yellow in color. And second, we have obtained 3 gametes which are yellow in color but they are wrinkled seed. They are 3 in number, again 3. They are round seed but they are green in color. And last we obtain that one is, that is small r is they are wrinkled and small y is they are green, wrinkled green, that is only one. So this is all about law of independence assortment or dihybrid cross, which is given by Gregor John Mendel, and this is essential in every exam they used to ask. First we have to draw the Punnett square and then we have to identify the gametes, their numbers, and by this way we will write their ratio. So this is all about your law of independent assortment. This is given by Mendel. Now one more topic we will understand today, and that is that topic is known as your sex determination. This is an important topic. Here how the gamete formation take place and what is the possibility of making male child and female child, sex determination in human beings, that we will understand in same chapter, that is principal inheritance and variation. So in this we will understand that sex determination. Here there are 2 types of chromosomes which are found. First we will understand that is called as homozygous chromosome. They are also known as your autosomes because they are similar chromosome. They are 23. Actually in human there are 23 pair chromosomes, but when we talk about autosomes they are only 22 pair chromosome, mean to say number is 44 chromosome. Now second we will understand heterozygous chromosome. Heterozygous chromosomes, they are also called as your sex chromosomes. That is one pair, they are one pair in number, one pair, mean to say 2 chromosomes, and represented by XX as well as XY. In a pair it is represented XY or XX. So how the crossing over takes place, this will understand with the help of a graph. Here the female is denoted with the help of XX gamete. This is the female parent. Female and male will be denoted by X and Y chromosome. We will make a cross in between and see what is the sex determination ratio we obtain. First we will make a cross like this way. First gamete, second gamete cross. Here we are obtaining X and X, then X and Y, X and X, X and Y. We will get the 4 gametes. XY is your male parent, and this is F1 generation. As we can see, here we identify that 2 female, need to say girl child, or they are called as daughter cell. So female gamete formation, that is X and X, and their percentage is equal to 50% because each gamete shows 25% contribution in this, as we can see. Now XY how much? 25 plus this 25 XY, and that is your boy formation take place, or male child we can say. male child, so that is your 50%. So in this we understand that 50% chances of a male child as well as which is represented by X and Y, and 50% chances of a girl child, and by this way sex determination take place. So in this we will take a different, as female have XX, that is mother cell gamete, and this is the father which have X and Y gamete. They are determining the parent, determining their genetic material, which you will see here 25% gamete of each daughter cell, where girl child getting 50% ratio as well as boy child getting again 50%. So we can say that genotypic ratio is also 50% means 1:1. So genotypic ratio in this, that is 1:1, right? And phenotypic also you can see phenotypic also 50%, and that you will see that it is also 1:1, because at 50% when you write like this, it will be, that you will cut it 1:1 that you are obtaining. Thank you friends.